Okay, so I am going to take you through a quick little lesson on blood types. You should already have this handout, and this handout has some space for some notes that we're going to take, and it also has the data tables for the lab that you are going to complete. So the first thing that I want to talk about is that there are 8 main types of blood. These are the main types: A, B, B, and O. Each type can have a negative or a positive, and the blood type is actually determined by a protein on the outside of a red blood cell called a marker molecule. So to kind of give you a little bit of background, I know you just read about blood, or you will read about blood in your student packet. Anything that I write on here, I want you to write on your paper. So a couple things I want you to know about blood. Blood is, it's considered a tissue on the tissue level of organization of the body. It's a mixture with 4 parts, okay? And those 4 parts are plasma, which is like the liquid part of the blood. You have the RBCs, which are the red blood cells, and their job is to transport the oxygen. And then you have white blood cells, and their job is to fight disease. And then finally, you have fragments called platelets, and their job is to help with blood clotting. You might think, well, blood clotting is bad. Well, right, you don't want a blood clot, in your circulatory system. But if you have a severed artery, or if you have like a cut on your hand, you want your blood to be able to clot so that you don't bleed out. This is kind of an interesting, it's kind of hard to see, but blood is constantly moving in your body. But if you were to take blood and put it in a vial and let it sit, it would settle like this in this mixture, and you would be able to see all 4 components. On the very top would be the liquid part, the plasma. You would have a little layer of platelets, a little bit of a layer of white blood cells, and then all down at the bottom, the most dense portion are the red blood cells. It would be a deep, deep dark red. But blood is normally moving, so this is all mixed together, but this just would be what blood would look like. If it wasn't moving, it would actually separate into these 4 components. So your blood type is a genetic trait. Okay, you inherit it from your parents. And we're going to talk a little bit about that. And what makes one blood type different from another is the fact that the red blood cells are a little bit different. So, a red blood cell, when you think about a red blood cell, it's just kind of like shaped like this. It's a red blood cell, and it has unique properties in and of itself. Its main job is to pick up the oxygen and take it to all the parts of your body. Now, red blood cells, what they didn't realize before they understood the science is that not every blood type can just be safely given to another blood type. So there are safe transfusions and there are unsafe, and people get blood transfusions all the time for lots of things. So some people have a condition called anemia where their hemoglobin is really low, and so they'll get a blood transfusion where literally their blood is taken out, someone else's blood is put in. But now we know that not every blood type is safe to receive and donate, so you have to be really careful. And so what makes it different are these little markers. So I'm going to try to take you through this little lesson here. So we're going to talk about each of the 4 blood types. So we're going to first talk about blood type A. And blood type A has a particular protein on the outside of it, a marker molecule. And we're going to represent it by like a little circle here. And its marker molecule is actually called anti-B. This will make sense here in just a second. So if I were going to draw a red blood cell that's blood type A, here's my red blood cell. This is blood type A. On the outside of my red blood cell, I'm going to have this protein marker, this antigen, and it's going to look, for example, like this. It's going to look like a circle. Now, it would just be a different chemical composition actually in the cell, but we're just simplifying it just with squares and circles to help us understand. And this is what blood type A would be. It has what's called an anti-B marker molecule. All right, then if I'm blood type B, okay, I actually have a different marker molecule. I have what's called anti-A. So I'm going to represent it here by like a square. So if I am drawing my blood type B right here, this would look a little different. My marker molecules look like this. So what this means is, for example, my red blood cells in my DNA If I'm type A blood, it's already coded for my body to make these particular types of antigens. And my body recognizes them, has no problem with them, is not going to see them as a foreign invader. If I'm blood type A and somebody gives me blood type B, and blood type B has these antigens on them, my body doesn't know what those are. It doesn't make them, it doesn't recognize them. So it almost sees it like a pathogen, like an immune response where it's going to try to attack it. If it tries to attack it, what happens is these blood cells can start to clump together. And as they're going through like capillaries, they can actually cause a clot. They could cause a lot of pain. It could even result in death. It could be a really dangerous situation. So in this scenario, if I'm blood type A, I don't recognize blood type B. This would not be a safe transfusion, and vice versa wouldn't be as well. Another unique blood type that's actually pretty rare is called blood type AB. And what's kind of interesting of blood type AB is it has two marker molecules. It actually has both anti-B and anti-A. So if I'm drawing this red blood cell and I am type AB blood, then this is what mine's going to look like. So my red blood cell makes both of these. So if I'm type AB blood, you might be thinking, well, that might be a good type of blood to have because I could receive from type B and it would be no problem because it recognizes type B. I could probably receive from A because it recognizes type A. So it just has two antigens. Then you have a special blood type. This is what Miss Barnett is. I'm blood type O. This has no marker molecules, none. So what this red blood cell would look like, it just lacks them. It has none. And we're going to talk about how that's significant. Now, one thing about your blood type, this is not like common knowledge. It's not on your birth certificate. The only reason why you would know your blood type is if you had to get tested for it. It's not like a common test that they do. So it's pretty normal not to know your blood type. I know my blood type because I am I'm not going to give it away, but I'm a blood type that a lot of people want for a different reason. And also in pregnancies, the blood type of my children was not compatible with my blood type. So all my children had to be tested. So I know all of my children's blood types too, which I'll talk about here in just a second. So it has to do with these marker molecules on the outside of the red blood cells. Another thing that you need to realize is that all blood types are either positive or negative. So you're either A positive or A negative. So you actually have 8 total blood types. Now the Rh factor is another protein and it's actually another trait. So this is a total separate genetic trait that you will inherit. It's called the Rh factor. If your red blood cell has it, you're called positive. If you don't have it, you're negative. Now, this may seem confusing, but I'm going to try to explain it. Rh positive blood types can receive a negative, but a negative cannot receive a positive. And this is why. Okay, let me try to see if I can draw to kind of show you how this would work. Let's just say that I'm type A positive blood. So here I am, I'm A positive. So I know that I have the anti-B markers on me that look like this. And I'm positive, which means I have this protein. I have this extra factor on my blood cell. So I'm just going to represent that just by putting Rh. So I am A positive, which means that my body makes the anti-B and the Rh antigens. And my body's totally okay with that. If I'm A negative, here's A negative. So I still have the anti-B marker molecules or antigens that look like this. And if I'm negative, it means I just don't have this. So I just don't have it. It's kind of like O not having it. It's just not there. So if you think about this logically, my body has to be able to recognize anything that's being put into it, like blood-wise. So if I'm an A negative and someone donates A positive blood to me, I am okay with these because my body recognizes them. Am I okay with the Rh factor? The answer would be no, because I don't have it. My body doesn't recognize it. If I'm given a red blood cell that has this factor, my body's going to see it as a foreign invader and try to attack it. Let's think oppositely. If I'm A positive and someone gives me A negative blood, I'm okay with this because I make them. And then A negative, it just doesn't even have the factor. So it doesn't affect this at all because it doesn't have it. So there's nothing to not recognize, if that makes sense. So it is safe for A negative to donate to A positive, but it is not safe for A positive to donate to A negative. Okay, let's keep working through it. Let's go to the back. All right, so how does this deal with genetics? Y'all remember the words genotype and phenotype. So two alleles you inherit are genotype, and we're going to be writing those down, and they all will have a physical expression of the blood type. Okay, so one possibility is you can inherit one big A and one big B. And so remember what capital letters mean? They mean that they are dominant. So this is actually a codominant blood type. So what's the physical blood type that you're going to have? It's actually going to be type AB. And I'm going to write over here that this is codominant. All right, another blood type. You could have blood type A. You might inherit two big A alleles. This would be type A blood. Or you can inherit one big A and one little o. So what does this tell us about type O? It's going to tell us that O is recessive. What happens if you have a capital letter with a lowercase letter? If you remember, the capital letter is going to dominate. So this is still type A blood. All right, you can inherit two big B's. Give me type B blood. You can inherit one big B and one little o, which also gives me type B. And the final one is you can inherit two little O's, which gives me type O blood. So type O blood is recessive. We are ignoring the Rh factor. Okay, we're ignoring it. But if I were to show you like how that works off to the side, okay, say this is a separate trait. Okay, say one parent is positive. The only way you can be positive is two positives, and one parent is negative. Rh is a dominant trait. So if I were to do a Punnett square simply like this, you can see that what would be the outcome of the Rh? It's 100% Rh positive blood types in this scenario. So that's just kind of interesting. That's just a separate little tidbit. So O is recessive, AB is codominant, and Rh factor is dominant. So let me show you like how would you do this like in a little Punnett square. Let me tell you a story. So my husband knew that his blood type was blood type A. Okay, and I knew that my blood type was blood type O. So I know that I am this because that's the only possibility. But my husband could either be this or this, and we didn't know which one he was. So guess what? We had our first child. We set up both Punnett squares. Okay, and because I told you that these are the two possibilities that my husband could be. Because my blood type was not compatible with my children, so they had to test all my children to kind of know their blood type. So we had our first child, and his blood type was type O. And so we were like, okay, so in order to have a type O child, this is going to tell us what my husband's blood type is because we knew that he was A. So if he's this type of A, if we work this Punnett square problem, is there a possibility of having a type O child? No. And we actually do have a couple of type A children as well. But if he was this type O, which means he's a carrier, could we have a child with type O blood? Well, I hope so, because we do. And yes, in this scenario, there's a 50% chance. So we were like, okay, so my husband has to be his genotype. Because we had an O child, so he has to be a carrier. So we kind of figured it out, by process of elimination, even though we didn't know his exact genotype. So sometimes you could ask your parents, hey, do you know what blood type you are? And you might be able to figure out your blood type by process of elimination. So I'm going to pause here in just a second, and I'm going to show you how we're going to set up our lab. Okay, so in just a second, once I explain this, you and your lab partners, you can work in groups of 2, 3, or 4, are going to carefully get a tray. On your tray, okay, you have the directions that basically I'm verbally telling you. These are all written down for you. Do not write on this. It says not to write on it right here. It says blood type lab, class copy, do not write on this. This is going to be for each class. You have colored water. They're all labeled, like green is AB, B is blue, A is yellow, and white is clear. You have this little well here. Each one has their own pipette. And then you have a toothpick. Okay, so you have the directions here that it's a class copy. On your paper, all you have are the data tables. Okay, so you have 1, 2, 3, 4 data tables. So I'm going to take you through one trial here to show you what you're going to do with this. And while you're doing this, you are recording all your stuff here. Excuse me. So you're going to be following this procedure 4 different times. Each time you're filling out your data table. Trial 1 donor type is type A. Okay, so we're going to do this together. So in my first data table, or I'm going to just show you, you're going to do this in a minute. I'm going to put type A as my donor type. You'll notice that I have all of the potential receivers. I have the original color, final color, and if it's safe or unsafe. So the directions say, first I'm going to place 5 drops of each blood type in each well. It should look like this. So I'm going to take 5 drops of AB, okay, and I'm going to say 1, 2, 3, 4, 5. 5 drops of B, 1, 2, 3, 4, 5, not contaminating, and then 5 drops of O. Okay, so there. So here are my original colors. So on my data table here, my original colors are always going to be the same, okay, for every trial. So you could even go and fill those out like this. You have the original colors, yellow, blue, green, and clear. Final color, and then safe or unsafe. So the next step says add your donor. For the first trial, the donor is type A. Add 5 drops of donor blood to each of your samples. Yes, even type A itself. So now I'm going to take type A. I'm going to add 5 drops. 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. And 2, 3, 4, 5. Okay. So now I'm going to mix it up. Okay, here's what I got. My final colors. So I would say my final color A, I would say that's yellow. My final color B, it is green. AB is also green, even though it's a different shade, and O is yellow. Those are my final colors. All right, so I stirred them with a toothpick to make sure that they mixed well. And then I have to determine if it's safe or unsafe. The donor is safe if the color remains the same. It's unsafe if it changes. A color change is an actual color change, like yellow to green, while a change in intensity, like yellow to dark yellow, is not. And then I have an example there of safe or unsafe, and I have an example of safe. All right, so if I were to determine this and I were to say, okay, A went from yellow to a little bit of a darker yellow, okay, that is going to be safe. My blue definitely changed from blue to more green, okay, so that is unsafe. My green stayed green, even though it kind of became a different shade of green. That is safe. And then my clear turned yellow, so that is unsafe. So in my first data table here, okay, I have my data. And you'll go back and do that. And then the directions say to gently wash out your Petri dish, repeat it, but you change the donor type for each trial until you've tested all 4 blood types as a potential donor. And then I have some reminders here. So I would go wash this out. So I'm going to go do that. All right, I've washed it out and I'm ready to do my additional trials. A couple of notes here. Your tray, when you're done with it, should look just like you got it, which hopefully is clean. You're never going to have to use a lot of this. Don't mix colors. Don't play around with colors. I have to have enough for all of my classes. So it should look like this, and it should be put back on the lab table. And you're also going to include the instructions with it so that that does not get messed up either, okay? Shouldn't have a whole lot of spillage. Be really, really careful. Once you finish, move this out of the way. Okay. So once you finish all of your donors, you're going to go through 4 times. You have some analysis questions. You should determine who's the universal receiver, universal donor. You've got some questions there. Questions here. Then there are questions here, 1, 2, 3, 4. You do not have to do 5. Okay, and it relates to this. And some people are like, I can't see. Everything is written off here to the side. The only one you might not be able to see is this is O negative 7%. Okay, everything else you should probably be able to see. This is A negative 6%. And you're going to answer these questions and you're going to see, okay, how common or uncommon these blood types are. Once you finish that blood type lab, you will turn it in. Okay, everybody has to do their own paper, even if you're working in a small group. You're going to turn this whole thing in. And then if you have time left over, you can do the assignment that I assigned, which is going back to that circulatory system gizmo. I have 2 more activities. There's another handout, but you open the same gizmo as we did earlier in the week, and you're going to do activity B and activity C. If you don't finish it, it is homework. Okay, thank y'all so much.